Good morning, everybody. It is 1:01 p.m. Hawaii time, so we need to get started here. And this session is going to be recorded for everybody's information. My name is Norman Arancon. I'm a professor of horticulture of the College of Agriculture, Forestry, and Natural Resource Management of UH Hilo, and I am the chair of Blake Miles' Thesis Committee. And I'm here to also introduce Blake before he speaks and delivers his defense for his thesis that he's been working on since 2020. So welcome everybody. For those who are here, I see a lot of very familiar people. Iching is one of our committee members here. Becky, Nat, Bob Peck from USGS, and Helen from USGS too, and we have other people here as well, so listening. Thank you for coming and sharing this momentous event for us, and especially for Blake. This is quite, what do you call it, something to be happy about, any defense for a master's student. So before I give the floor to Blake, I would like to introduce him and where he's coming from and what he's actually has done for his thesis. So Blake Miles is a fourth-year Master of Science graduate student at the University of Hawaii at Hilo, participating under the Tropical Conservation Biology and Environmental Science Master's Program. Thesis track: born in Honolulu, Hawaii, and raised in San Jose, California, Blake's admiration of biological sciences, especially insects, developed early in life. Blake received his bachelor's degree in entomology, Bachelor of Science degree in entomology from University of California, Riverside in 2017. And during his attendance in that university, Blake procured an interest to pursue a professional and academic career in entomological invasive species management, which continues to this day. For the coming future, Blake is currently seeking admissions into a doctoral program in entomology. Since fall 2020, Blake's thesis research project for the TCBES MS program has focused on addressing scientific knowledge gaps relating to invasive Queensland Longhorn beetle, in short QLB, populations localized in Puna District, Hawaiʻi County. Without much more ado, I'd like to give you now the floor to Blake to tell you more about it. Blake?
All right. Well, then let's get right to it, shall we? Let's see. Alrighty. Thank you. Thank you so much. As you've heard, my name is Blake Miles. Thank you for attending my thesis defense. And today I would like to talk to you about the characterization of Queensland Longhorn beetles in Puna District with regards to their distribution patterns, host selection preference, and environmental correlations in Puna District, Hawaii County, Hawaii. So this is going to be our presentation outline for today. And without further ado, let's move on into introductions. So for many insects that are invasive to new localized areas, we at times may not know much about their capacity to disrupt their environments. And so when it comes to this, gaps in knowledge, a good place to start would be to better understand how deep their host preference range encompasses, as well as their behavior in host seeking. And so this is, for an example, a series of types of true weevils that are called ambrosia beetles, where we have, among many genera, they are inclined to infest deadwood, but in other species, they tend to infest living host plants, which can imply various implications depending on the ecosystems in question. To your right are a number of scoletinid examples of species. So host preferences among various insect species can also vary depending on, given the climate, the elevation, the latitude, and the geographical ranges as well. And suitability for suitable host availability can in turn significantly influence their reproduction. So how many generations can they sustain in a year, their breeding success, their volatilism. Let's see. And for example, we have a notable pest insect called Asian longhorn beetle, which is in the subfamily Lammiinae. And we have a study that was conducted in Europe, where in Italy, they were found not to target a certain type of tree called green ash trees, but whereas we have populations of ALB in North America where they go crazy over that. And so without sufficient access to region-specific studies for unpredictable species like Asian longhorn beetle, designing management and pesticide protocols can prove difficult. And the pictures provided in this slide indicate infestation, serious infestation of pine trees from different types of bark beetles. So this takes us to our topic, our main insect for today, QLB, Queensland longhorn beetle. This is also in the same subfamily as Asian longhorn beetle, coincidentally. So as a recent invasive species to Hawaii, it was introduced into Queensland from Queensland, Australia. And prior to its introduction, no one paid mind to it. It was never economically or commercially significant to warrant a closer look. And so in terms of a developing population, the first sightings of an adult specifically were made in July 2009 in Hawaiian Acres in Puna District, Hawaii County, where we are. So a couple of years would pass, not really much of anything going on until you would find gradually over time, more and more adult beetle sightings being reported, such as in 2013. And then the next year in 2014, they had a first confirmation of a host larval association with a weakened breadfruit plant. They found larvae boring into a dying like trunk of that species. And then as of 2020, larval infestations are reported to be present across various locations in Puna District. So in terms of targeting, QLB is regarded as prolificous. It might go after a wide suite of different plant species, ranging from between agricultural or ornamental, culturally significant species in Puna District. And as far as what we know about what's targeted, we have confirmed through BISC, through Hawaii Department of Agriculture, that 10 plant species are that are confirmed so far to support full-term development and egg laying. We have 3 species that are still speculated. Additional data and rearing is required to confirm these. And from looking at phylogenetic tables such as this one to your left, we can say that they're not, the majority of the species are not related closely. So this is for just going through what we know. We have Queen sago palm that's confirmed. We have Norfolk pine, cut logs. We have avocado, breadfruit, trumpet tree, kukui, cacao, citrus, and calamunge. And for speculated, we have naturalized croton, gunpowder tree, as well as hibiscus plants in that genera, hibiscus species. So as far as the physical characteristics concerned for adults, they are quite plain with a dark brown coloration. They are ranged between, their lengths go from 2.0 to 4.5 centimeters and are believed to forage. Their feeding habits are not known, but are speculated they could be feeding on host bark, leaves, or petioles, but they don't cause as many complications as do their larval counterparts, which are very much vermiform, legless, large-headed, cream-colored. They're longer in length than the adults, usually from 5.75 centimeters, and their damage comes from boring into tissues for sustenance, where they form galleries. So how do we determine or spot signs of infestation? That goes, a number of characters can include oozing sap around egg-laying sites made by the females. There's trunk girdling, sawdust like frass accumulation, and there's also adult emergence holes that are about 1.25 centimeters in diameter. The consequences of larval infestation can be minor to grave, with compromised host health for starters, which can segue into secondary pathogenic infections. In more advanced stages, you'll see branch dieback dropping, and in worst case scenarios, death in plants that are significantly compromised and they're sustaining heavy infestation, as you see here in the image to the left, the right, pardon me. So what do we don't know about QLB? Well, virtually lots of things about the majority of knowledge gaps encompass the biology, ecology, life history, and behaviors. There was a study that was conducted in 2019 for testing response behavior to cerambycid aggregation sex pheromones. Other than that, we are still learning more about the species, and progress thus has not been made yet in developing management control techniques or identifying insecticides that would be effective for proper management. So for the cultural risks, if this population of QLB goes unchecked, it could very well threaten Hawaii's cacao industry, as well as the citrus industry in the United States, that it's an annual worth of $3.85 billion. That is provided if it manages to establish in Florida. So in October of 2019, we had a United States Senator by the name of Brian Schatz, who urged for collaboration between both United States federal departments and private county state landowners to come up with solutions, figure out more about how QLP operates, and as well as with the management development of those protocols to manage the species. So in terms of comparative referencing, we could use a related tax such as Asian Longhorn beetle as a possible springboard to test preliminary hypotheses and management protocols. Very, very, very much just preliminary, if you will. That includes biological aspects that could overlap with traits, as well as with chemical control applications, such as systemic insecticides, then considering are there any similarities with olfactory visual responses to plant motifs as well. So under ideal circumstances, if we were to consider a push-pull management plan, it would deter QLB away from preferred target plant species and instead to redirect them towards traps that can be managed. So this takes us to our objectives for today. And I want to just let everyone know that as part of a bigger United States Geological Survey and Hawaii Cooperative Study Unit research project, this study sought to improve baseline knowledge of our populations of QLB in Puna District by first characterizing, assessing the current distribution patterns, identifying host selection preferences, as well as identify environmental factors that could be correlated with infestation activity. So now we will move on to our materials and methods. For starters, we took to collecting sighting records obtained from the Big Island Invasive Species Committee, Department of Agriculture in Hawaii, as well as with the United States Department of Agriculture itself, federally speaking. And in terms of our survey areas, we confined all of our survey area to the Puna District with what we collected data-wise from reports. And this took us to sticking to sites that were in East Puna, Kau, Curtistown, Mountain View, and Bahoa. So in total, 20 of those sites were surveyed. All of them were private. So we had about 16 residential properties, 3 orchards, 1 farm. Every landowner was notified and given consent prior to surveys. And in terms of disclaimers, the method sampling protocols were adapted from a study that was on gold jackal den selection. We would have used a study that compared similar taxa to QLB, but the methods that we found were not compatible or necessary, such as in the case of Holker et al., we had tree felling, we had too few survey sites, and samples were collected for processing in the laboratory. This was all in the field, what we did. So the survey duration took place from October of 2020 to March of 2021. And on average, it would go, individual surveys would last from an hour or 2 hours at least. All sites were visited once, whether or not QLB was confirmed present and absent. And despite the range phenology, despite that not being well characterized as of late, Larval infestation is believed to still be visible year-round. Vegetation sampling was also restricted to the immediate property areas. We didn't go anywhere out of their areas. And so for each survey, all tree-like vegetation was sampled, regardless of known host stamens. So anything that wasn't reported yet, as confirmed, speculated, we just sampled whatever we can get our hands on. That excluded shrubs, ferns, epiphytes, and smaller herbs. Property owners were instructed not to reveal locations of plants suspected of harboring infestation. And as surveyors visited the sites, we began at site entrances for sampling and eventually worked our way around the properties until coming to the opposite end. So for our data collection in terms of sites, we took everything from the property names, what types they were, from their GPS coordinates to the surveyor names that were running the surveys themselves. Plant-wise, we also characterized their identification codes of unique plants that we sampled, took measurements of their trunks, main stems, and including any GPS coordinates of plants recorded to be infested with QLB. So for both survey data, after we did collect our data from the field, we took time to extract information relative to topographical and climatological metadata, which broke down to at least three items of interest: average elevation, average annual air temperature, and average annual rainfall. So in choosing plant species to analyze, we, over the course of the sampling duration of 6 months, had a lot of species that we did inspect. But for our ease of analysis and in terms of providing good data, we filtered any species out that were surveyed less than 15 times, and we filtered out any that had less than 5 individuals infested with QLB. So that resulted in us conferring these 5 plant species for our study, which is kukui, breadfruit, trumpet tree, citrus, and cacao. So after with our selection, we devised botanical descriptions to provide a better context of what kind of plants that we're working around with. So that is just a breakdown of the generalized characters, examples of infestation, growth behaviors, not comprehensive, but just something to get a general idea. So we'll start with kukui. It's a Polynesian introduction from Malaysia to Australia. It's been a state tree in Hawaii since 1959. And it's quite a hardy little plant, tolerant to drought, salt spray, and has had various applications in Hawaiian culture, such as use of oil for candle illumination and canoe crafting. These are some example insect species that are known to attack kukui in Hawaii. We have black twig borer, and we also have Magnolia white scale that feed on leaf tissues. The growth habit that kukui assumes is a spreading tree. It has a variable leaf shape, and the seeds of the fruits can be mildly toxic when raw due to foreballs and saponins. And for our next plant is breadfruit. This is also another Polynesian introduction from Bismarck Archipelago, possibly in that range. It's easily grown and managed in various areas, but its use has declined since the 1970s and 80s. It also has had similar multi-use applications in Hawaiian culture. So these are also examples of tree pests to breadfruit, such as barnacle scale and spiraling whitefly. And this is another spreading tree that assumes that kind of growth habit. It has a dense canopy. Its leaf features are generally consistent, clustered at the ends of branchlets, pinnatifid, and the fruit features are slightly bumpy to spiny, broadly cylindrical to globose. This is a type of pioneering weed tree, trumpet tree, rapid growing, introduced from central south to part of Mexico. And it was introduced to the Panaiva Forest Reserve as a watershed cover. And so interestingly, in its native range, it does form mutual associations with a genus of ant species called aesthetica. And this is actually in its range of Costa Rica. It's known to be attacked by brown twig beetle, another type of ambrosia beetle in the genus Xylosandrus. So this is a spindly growth tree that's evergreen. Trunk diameter is quite narrow, 20 centimeters, and generally its leaf shape is consistent. but it's pinnatifid branchlet spreading, and its fruit features are also achenes. This is citrus. It's durable too, just like kukui. It can grow in compacted soil. It has a high sensitivity to pests and fungal diseases, and it's got a bunch of various types of cultivars under its genus. These are also examples of pest insects that can attack citrus in Hawaii. We have black twig borer, citrus leaf miner, and Asian citrus psyllid. And this is another tree with a evergreen growth habit, resistant to breakage. Its leaf features are variable, and it has, for its fruit, it can be round with a fleshy covering. And this is cacao energy. This is part of an emerging commercial industry in Hawaii, introduced during the 1850s, and it has a worldwide market demand that generates about 75 million USD annually. And its raw materials can be at times harvested for consumption and use of multiple product generation. And these are also examples of pest insects that attack cacao in Hawaii. We have Chinese rose beetle and brown twig borer. And this is another evergreen tree growth habit with a height measurement from 4 to 9 millimeters. Its leaves are consistently simple, lanceolate, and the fruit consists of hard shell pods where in each they contain around 20 to 50 seeds coated in a creamy pulp. Here we go. So in terms of our data analysis, because that's what we'll segue to now, we used Google Earth to create distribution pattern maps for all of the site infestations of QLB and Puna. Very basic, but we made maps for each species, including one that lumps all of them together. And the coordinate data was, we took care to mark each of the data by specific markers. And so moving on to our preferred host analysis, we used RStudio to conduct mixed effects logistic regression modeling on all of our host selection preferences for the population. And our statistical methods were adapted from Manley et al., which is viable for any studies, ecological or biological, that measure food and habitat selection in animal populations. So this is just a run through of our summary protocols, not comprehensive, but it gets the idea across. We imported data, we filtered it to exclude fewer than 15 instances surveyed for any species. We characterized, did a first attempt with generalized linear modeling, where through that we assigned kukui as an intercept. And we then, just to make sure, did a linear relationship assessment of tree size variables. We decided to remove plant height as a predictor variable due to its high correlation. Usually keeping correlative values, coefficients between 7.0 is ideal. And then we had, of course, created the generalized linear effects mixed model, and then led to post hoc testing with 2D contrast, and then did a diagnostic at the end. So for our final segment of the objectives, we had environmental infestation correlation. So we just compared all of the tree species to individually average elevation, average annual temperature, and average annual rainfall. And this was all done through Pearson's product moment correlation test. So this is just another walkthrough. Import, filter by census areas, run the Pearson's test, but preliminarily we determined that average annual temperature and average elevation were strongly correlated, and did everything for the aforementioned. And so this is for our results for today, and we will go through this. And this is a map of all of the distribution patterns of QLB, just a sum, where out of a total of 1,087 trees sampled, we found that 216 of them turned out to be infested, which equates to a infestation rate of about 19.8%. This is also a map specifically for kukui. We had a total of 34 kukui sampled, and out of those we had 23 that were infested, and that equates to about 64.7% infestation rate. And as for breadfruit, we sampled a total of 35 plants, and out of those breadfruit, we had 8 that were infested, which equates to 22.9% total infestation rate. And this is for trumpet tree for the distribution map. We had 85 that we sampled. Out of those 85, we had 14 that turned out to be infested with QLB, and which amounted to 16.5% infestation rate total. And then for citrus, we had quite a bit that we surveyed, about 160, yes, 160 if you will. But out of those, we had 19 that were infested with QLB, and that was equates to 11.8% infestation rate. And finally, we have cacao infestations. Out of 772 that we sampled, mainly on orchards and farms, we had 152 that were infested, which was equivalent to 19.7% of those being infested. And so let's talk now about the preferred host plants. These are differences in mean QLB infestation coefficient comparisons, but out of these, we found that in fact, we had significant data with regards to kukui. It was found that they were preferred over infested trumpet tree, citrus, and breadfruit, and the same also applies to cacao, where cacao is also preferred for infestation over trumpet trees, citrus, and breadfruit. And so lastly, for our third item for objectives, we have environment infestation correlations. And in summation, we did not find any correlations observed through Pearson's product moment testing. For kukui, that is also the same, no significant correlations. For breadfruit, what we did find was that there were strong, positive, significant correlations for both mean elevation and mean annual rainfall, respectively. And then for trumpet tree, we did not yield any significant correlations in the data for any of those climatological factors. And then lastly, citrus, well, second, penta-ultimate. Citrus, we did not have any significant correlations observed either. And as for cacao, we did find that there was a strong positive significant correlation between cacao infestation and mean annual rainfall. And so now we will move on to our discussion interface. And this is where I'd like to talk a little bit about what we found from the data. So we did again say that we did not find any significant dispersal patterns observed across the sites in Puna district. And we have a bunch of ideas as of potential influences. It could range between, could it be climate? Could there be biological influences, geographical areas that might be influencing their distribution, their behavior for lack of? Is there anthropogenic causes? Of note, opportunism and travel path, what's that? Well, for a number of species like Asian longhorn beetle, we know that if there is a suitable host within its range, ideally, not well, it will just by happenstance be able to go there, you know, to whatever it can reach. And at times it might span between travel path-wise, walking or flight, and it may not always be direct. It might take time before it reaches its target host specifically. And what about host condition? That's also could have possible implications, because that could say for a tree that might be susceptible, we have to consider its physiology, maybe how old is it, you know, and if it's under stress, what kind of stress? And that also goes into, as a result, stress emissions. For example, if we were to consider it general environmental stress, at times, well, metabolic production of certain chemicals such as ethanol, acetic acid, and methanol can increase. And those in turn could be potential cues for attraction. Another interesting point that we could make about about possible influences are aggregation pheromones. For many of the species and Lemmiani longhorn beetles, the pheromones are actually male produced exclusively, but at times there can be occasions where the pheromones act synergistically with tree emissions, phytochemical emissions that are produced that create a stronger pull, like a attraction to where they need to go for mating and spreading their progeny. So that's one consideration, but it's we need, as time goes, we will learn more about their biology and behaviors in terms of distribution patterns. So now let's talk a little bit about the preferred host plants. Significantly, there is a host preference for kukui and cacao plants, respectively, over trumpet tree, citrus, and breadfruit. And two items of note for potential influences could be the case for the types of chemical profiles. Very rarely are they individual. They're a lot of times consistent of blends in phytochemical emissions. So we have two major classes that seem to be attractants for a lot of destructive beetles. And we'll have to say, for example, there's terpenoid metabolites, which are lipids, big group. Some are insecticidal, but of course, you do get a blend of those where they can prove attractive to Asian longhorn beetle yet again. Then there's other types called phenolic metabolites where they're aromatic in character, another large group. And same thing as with terpenoids, some are insecticidal to ward off predators. But some insects like emerald ash borer, which is also potentially big insect invasive, is able to avert these defenses by detoxifying and excreting the phenolics that are contained in ash trees. And so it depends on the insect species, but potentially there could be a certain blend or types of compounds that interact with each other to create the reason for why QLB would be inclined to attack cuckoo in cacao. Too early to say. Another potential influence could be the attraction to chemical profiles that are actually tissue specific. Because in one case, we have breadfruit where the leaves themselves are known to accumulate phenolic anthraquinone products, whereas the bark itself specifically concentrates different types of flavonoids as well as with dye and triterpenoid compounds. So that could be interesting if maybe perhaps there might be some considerations with growing seasons where you have fruit produce being grown, so just speculations, but nothing conclusive. But now we also have, finally, for our discussion, environmental infestation correlations, where we did find that cacao infestation correlates significantly, positively and strongly, yet again, with mean elevation and mean annual rainfall. And so for the plants themselves, their ideal rainfall, the rainfall that's suitable for their development ranges between 3,000 to 4,000 millimeters annually in regions where they're grown. And elevation-induced temperature stress should not be any kind of issue for the plants themselves, because throughout the surveys that we've visited for properties, elevation did not exhibit significant temperature differences. So that's elevation-induced temperature stress, mind you, if I botched that before. So for the infestations of cacao themselves, they could be possibly influenced by mean elevation in conjunction with mean average rainfall, but the caveat being variably, though, that might depend. So with breadfruit, it is of the same infestations are strongly and significantly with mean annual rainfall. So in their ideal ranges for where they're grown, they are happy to receive anywhere between 1,500 to 3,000 millimeters of rainfall average annually. But for both cases of breadfruit and cacao, if infestation is mediated by precipitation stress, getting too much water, flooding is not likely going to be an issue for these plants, because with the way that, geologically speaking, that the islands consist of soil and volcanic rock, their hydrology actually allows for extremely high permeability and infiltration rates, meaning that you're not going to be easily getting floods unless there are severe storm periods where they last for various amount of days. And so just below is just to show you the average annual rainfalls between our sites by census area to give that context. So for our retrospections, I want to just go over for future studies because it is important. And just to make clear that this study was limited. We only had 6 months of survey period observations. For the future, it would be great to have it all year round so that the full phenology, the potential scope of reproduction, breeding, and development can be observed in the long QLB populations in Puna district. But also a big caveat is that the protocols that were used for creating distribution maps was very basic. This was all just a baseline assessment of what we're seeing. So therefore, we did not utilize sophisticated algorithms, mapping programs such as ArcGIS, but of course, with future studies and as we accumulate more and more data on the longhorn beetle's ability to infest, that invites more sophisticated software. So we also have a limitation in assessing the host selection preferences, and that could be, that was also due to a lack of Puna district specific density data for different cultivars, plant species abundances. Otherwise, we would have applied that to good effect. And lastly, for just the purposes of this presentation, we did have sites that were featuring high heterogeneity. So that's multiple different plant species in one area. There are plant species that had high densities, low densities, very variable. So as the sites were variable, that could have also had a hand with impacting the host travel, search time, just the behaviors of the Queensland longhorn beetle, similar to Asian longhorn beetle itself, ALB. That's why it could be of importance of consideration to consider comparing traits between analog species within subfamily range, even perhaps comparing them between different subfamilies within the order. So in conclusion, I want to just go through as just to take away from all this, as I appreciate your time. For our conclusions for objective 1, we found that there were no observed significant distribution patterns of Queensland longhorn beetle infestation across the sites in Puna district. So it's there was no significant pattern at all. For our second objective, we concluded that host selection behavior among Queensland longhorn beetle population significantly preferred the infestation of kukui plants over the infestation of trumpet tree, citrus, and breadfruit. And this also applies the same to cacao. Infestation of cacao over trumpet tree, citrus, and breadfruit. And for our final conclusion for the objective, breadfruit infestation in Puna district was significantly correlated strongly and positively with mean annual rainfall. And as with cacao, infestation of the plant in Puna district also significantly correlates strongly and positively with mean elevation and mean annual rainfall. So I would like to give my acknowledgements, as there are many to thank for their time generously in this process of coming to this point. I'd like to thank my thesis committee, everyone, especially Norman Aricon, the chair of the committee, for his time, advisory, and support, especially with the data, the check-ins by week. I would like to thank Drs. Cam Muir and Eugene Lee for their participation support. And as part of the USGS Pacific Island Ecosystems Research Center is concerned, I have my many thanks to give to Helen Sofair, who was very generous with providing resources, as well as with data analysis assistance and overall support through the process. And in terms of the Hawaii Cooperative Studies Unit, situated at University of Hawaii Hilo, I have also my many respects and appreciations to give to Robert Peck for resources as well as with support through the study. There's also Jordan Zarters, who I have my many thanks to give for survey coordination, data entry, and just his support too. And for the Peer USG Center, I also thank the federal grant funding support that was made possible through the USGS side of the QLB project, which is co-managed by PIs Helen Sofair and Robert Peck themselves. We do also have the Hawaii Cooperative Studies Unit at University of Hawaii Hilo, who I would like to especially thank Sharon Ziegler-Chong, the director, and Lori Buffel with us right now as project coordinator for their for student employment, hire, and support. And as for the case of just in a generality with the University of Hawaii Hilo, I would like to thank Tracy Wagner and Rebecca Ostertag for both their support and direction in my master's thesis. And then just overall, just my major appreciation for being able to give them a chance to take on this challenge and just achieve the goals that are laid out. And lastly, I would like to give thanks to my family who helped encourage me to keep going and to adapt and overcome and persevere through times of, but it's just adapting and overcoming. And I have my endless thanks to give to all of them. And so just for lastly, if anyone has questions regarding KLB management, well, just not management, just any contact questions, I would like to give my email address and for any questions that would like to be answered. Thank you. Thank you very much for your time, everyone.
Thank you, Blake.